subgroups is, even though I believe those varying effect models are smooth, I believe the truth is usually smooth underneath it all. You have to design a study that either has somebody in it or doesn't have somebody in it. So that's why I present things in subgroup-centric sort of ways. And so we'll think about those varying effect models in that way as well. So basically, ultimately, at the end, you're going to have to describe a group of patients you're going to study. You're going to have to specify some model for the patients in that group. This is sort of an average model for everybody in the subgroup. There might be a smooth underlying model generating this, but that's describing what's going on with the patients. And this very important function here, it looks nice. It looks like might be real mathematics because it has an integration sign there, but it's really not. What it is it says how many people or what fraction of people are in that group. So these are clear functions. Everything that comes to my mind when I'm helping somebody design a trial. I want to know the effect. I want to guess at the effect in the group, but also be able to tell me how many people I'm going to be studying. And so methods ultimately that help me describe the patients that are on the study, tell me the model for the patients that are going on the study or the effect size, and also tell me what fraction of patients in the group are really, that's the triple that's important. And these might be models to represent that. So not to spend too much time, but so underlying all of these ad hoc methods, that's kind of what's in my head when I'm doing them. And this slide I put in 2 days ago. I promised myself I wasn't going to talk about trees and subgroups, but this is mostly for Sunil's sake and others since I played with this so much. If you get to that point in time, then there are tools available to help you describe those boxes, and these are tools that describe these subgroups that you're having to act on. The most famous being regression trees and survival trees, which essentially partition data and give you these rules. More recent ones that we worked on for SNP data that have binary things called logic regression, but essentially give you rules like this. And the most recent one, which nobody seems to like, even though I thought I gave it a great name like extreme regression, but it gives you rules like this as well. And so you can Google search those ones, and they're probably up on my website. But those, if you get, this is sort of, if life was so good you get to that point, these are the places maybe where you should go. But what I'm going to spend the remaining part of my talk talking about is variance control, which is the bottom of this thing. To get there, you have to be able to pick the variables you have a chance of finding the interactions in, because these are very adaptive procedures. And as great as these adaptive procedures are, as you can see here, they're quite discrete. They look for cut points and things like that, so you need to sort of control the variance going in. So go simple. This is the message I'm going to try to convey. So how were some ad hoc ideas we've tried in the past to do this? I agree, there's probably much more principled statistical ways of going at this, and several in the audience would go there. But since I'm a pretty applied statistician, I'm just going to talk about some ad hoc ways of getting there. The first one being, as I said before, look for lower order things before you look for higher order things. But that's sort of a traditional statistical, that's part of our statistical bread and butter. Don't go for something more complicated before you can find a simpler solution to your problem. But this technique is sometimes useful in the sense that the higher order question has a lot more dimensionality, so it's more of a shifting thing, of trying to keep filtering thing. Number two, which relates to clinical trials a lot, and I'll spend a bit more time, is if you think the treatment really does vary with respect to that marker, then build that into your design, build that into the look for it, Commit yourself to a little extra looking in a well-controlled way. And finally, if I do get a chance to do that, since I probably will run long, is saying, what happens if you do that too fast? So those are the contributions. So what's strategy one? One is to focus on interaction, but only consider those variables that are most promising. So do a filter, but do filter on the main effects. And it's easiest to think about this in linear regression. It works for logistic regression, for binary data, for survival data. It becomes a little bit more complicated to keep everything the right. But in the linear case, think about a model that involves x1, x2, x1, and x2. So we'll keep it completely generic. And a main effect model that many of us statisticians might fit if we're interested in a single variable would be a model in x1. That might be the first thing we'd fit. And then also we'd be interested in this interaction effect. The thing to take away from it is this estimate here and this estimate here are essentially uncorrelated. And so you can construct all sorts of test statistics where the first look at the data is uncorrelated with the second look at the data. And what that means is you can filter 1,000 things to begin with, and then look at the 10 most interesting ones marginally and say, oh, I'm going to test interactions in those. But statisticians always like to do Bonferroni collection and things like that, or some version of, you only need to control your type 1 error for the second look. Kind of makes sense. The first one's independent, the second one's pretty easy. And you think, oh, this must work in general. But it actually takes a little bit to actually get the proof through. And this is James Dye's work, where if you construct all these estimators and they're normal, Gaussian, which most of them are pretty much, then if they're uncorrelated as the sample size gets big, this arithmetic says, and this is the second stage interaction testing, is down here, you look, here's the alpha divided by some number. It says, oh, I only need to divide that alpha by M0, which is the second stage number of comparisons. So you get to look a lot for free on the first stage as long as you specify in the second stage. It comes at a price that if you're really unlucky, if the gods are malicious, you will have missed those interactions because they'll be perfectly hidden. But if things are not so bad, you might have caught some of them, and then you don't need to pay for that second look at the data. So this actually is nice because it doesn't, the filter couldn't be not just on a main effect, it could be any sort of constructed sensible orthogonal statistic at the first thing. So that's one of the things we've done to try to limit dimensionality in looking for interactions. The other one is, and I'll spend a few more slides here, is one where you say, oh, I know there's a subgroup, let's go for that subgroup first. So these are some examples of associations I might be looking at. I might be interested in genetic associations with survival. I might be interested in severe toxicity markers. Those are main effects, but can I find those main effects? Help me find them by looking for a subgroup effect first. Or I may also be interested in interactions, and those second bullet points are interactions. The last one, say, for instance, is there a genetic association if only in a subgroup of patients? So for instance, suppose the association is only in smokers, and now I'm veering away from my clinical trials examples into my colleagues in genetic association studies. But the same idea, if the effect is strongest someplace, I can't really know exactly where it is, but if I have an adaptive algorithm and find it in a well-controlled, variance-controlled way, that might help me do some filtering. So this is the picture. I think my simple schematic, my limited PowerPoint drawing will take me there. It shows 4 boxes, and it says, suppose there's some weak marginal effect. And so think about the yellow and blue representing response from therapy for these two groups of patients. If I could throw away some of the data or downweight it, I might see a stronger effect. If I could throw away more of it, I'd see a stronger effect. If I throw away more, I'd see a stronger, the intensity being how strong the effect is. Now, you know, those of us working in clinical trials in oncology, we've done this before. We've been pulled into this. No, I know there's a subgroup of patients where the treatment works. I told it. That's really an okay thing. I was too resistant to this at first. You can do it, but you just need to be controlled. You need to understand the algorithm you're doing it. And so there's weak marginal association, but enhanced association with subgroup. That means if you're just interested in seeing whether it works in general, there are better ways to go look for that signal. So in terms of arithmetic, what am I talking about? Well, statisticians, usually all of our test statistics really look like sums of things. And so here I'm writing out score components. And really what I'm trying to say is here, if I could, the standard statistic might have all these h's and 1s. So basically think about this as a function that depends on some other variables and some parameter of interest, and these are the score components of the test I would do generally. It might be part of the log rank test between the two survival. It could be part of the binomial test that compared these, and there's a component for each variable. The message is, can we plug in some other number here rather than 1 to weight the right thing? And the answer is yes, you can. So if you think about this slightly more generalized version, we might have functions that depend on different clinical factors or genetic factors. We might have parameters in front of that stuff, and we're just going to weight these guys to make the test statistic standardized as big as possible. So this might be the marginal test, not doing anything like we do. This might be the test statistic when we say, oh, I'm going to let the data guide us a little bit on one variable. It might be expression, EGFR expression. And then this might be the test statistic. I'm not only going to weight it on that, I'm also going to weight it on some of their other clinical characteristics. And so there's a whole statistical world of boosting, but you can kind of think of this as boosting in a different way. You're directing your boosting right at the interaction, not at the overall prediction. Boosting's been very popular in thinking about improving my prediction rules. Think of it as a, you know, off-the-shelf, making everything better sort of algorithm, putting it that way. But here what you're trying to do is you're focusing it just on the interaction or the subgroup. So that's some of the work we looked at there. I will say in a more principled statistical way, rather than thinking about test statistics, I might have formulated this as a model that looked like this, where it involved some covariate factors and an interaction. And here I'm talking about gene by exposure variable interaction, not treatment, but some complicated function involved those things, and I'm just doing it in a very organized, estimating way, and sort of profiling out this solution. So there's sort of an ad hoc version, and then there's a model, but most of them I'm driven by also how fast I can actually program it at these ages as well. And on top of that, so I said boosting, but of course there are ways of getting almost there by doing more efficient methods. And so we actually end up doing estimation using a very popular technique that many of you have heard about before, using L1 type regularization. And in fact, the algorithm we use is related to that in some respects. It's a Lars-like algorithm. But basically, it's a fast way of doing, for us, it's a fast way of doing boosting in a way we can focus on the interactions at hand. And the final thing, as a statistician, you always need to know what the p-value is. No, you don't always need to know what the p-value is, but some of us use it as a good summary statistic. And so these very simple, the advantage of working in simple statistics is sometimes it's also easy to do calibration for what error is. And so essentially we construct these by adding a little normal noise on it.